traits like. Now, this is an extreme example of population adaptation to a predatory lifestyle since it resulted in full speciation, but in many other species, studies have investigated the interaction between adaptation to localized prey availability and population level divergences. All of these studies note how specifically a generalist predator can, when introduced to novel ecological niches, undergo adaptation should gene flow be limited by some mechanism. Thus, there are three major ingredients, so to speak of, that can promote population and then eventually species level divergence based on adaptation to predatory lifestyle. These are one, a relatively novel environment with ecological opportunity, two, a generalist diet within the ancestral population, and finally, three, some form of limitation to gene flow within said environment. North America, as a geographic area, provides one of these key ingredients naturally due to the recent retreat of glacial maxima. So, glacial retreat in the area, specifically along the Atlantic coast, provides a perfect environment for looking into species range and ecological niche expansions into a novel environment. This is largely due to the glacial retreat out of the area, specifically occurring between 15,000 years ago. These environments are still relatively young from an evolutionary standpoint, providing opportunities for investigating eco-evolutionary dynamics like population adaptation to diet. But this is only one of the three key ingredients needed for adaptations to predatory lifestyle to meaningfully influence species populations. You still need to identify species with generalist diets that can promote this diversification, and you perhaps more crucially need to identify candidate species that have some form of mechanisms that restrict gene flow in between their populations. This last point is typically viewed as the limiting factor in this equation, specifically regarding marine fish populations. It's one of the more well-established phenomena that marine fish are some of the most panmictic species on the planet. Many species occur over massive geographic ranges and possess great dispersal capabilities, which makes population differentiation like all but non-existent. This is a study from 2023 that looked at population genetic structuring of American eels across much of the Western Atlantic from Canada to Trinidad, revealing a complete lack of population structuring over such a massive geographic area. Another study looking at whole genome-based genetics structuring in sablefish within the northern Pacific Ocean has similar results. Again, a complete lack of genetic structuring and evidence for intense gene flow between locations where specimens are captured. But this isn't always the case. Shown here are the results of two whole genome level population genetic studies pertaining to corkwing wrasse on the left and gold cine wrasse on the right. These studies found that across the northeastern Atlantic, where these two wrasse species are endemic, populations show evidence of structuring over fine geographic distances, sometimes in as little of areas between southwestern and southeastern Norway. It's becoming clear that species life history and dispersal capabilities infer the connectivity of populations in marine environments, and that despite the mass amount of environmental connectivity present in a marine ecosystem, species can still demonstrate fine-scale structuring that can help promote adaptation. And so, should we be able to find a fish species with a generalist predator lifestyle, that B demonstrates population structuring within C, our novel environment of the northwestern Atlantic, we may be able to look for signatures of adaptation to dietary regimes within distinct subpopulations. And lo and behold, would you look at that, we have a perfect candidate. The Cunner Wrasse is another wrasse species endemic specifically to the northwestern Atlantic, overlapping its range with the same environments that were only recently deglaciated in the past 15,000 years. Cunner have also demonstrated to be generalist predators of benthic invertebrate communities, presenting opportunities for dietary differentiation in this species, especially if the available prey between different regions where the species is found can vary. So, two key ingredients are present here. And third, and perhaps most importantly, there is evidence that gene flow is limited and subpopulation structure exists in this species, as shown by this study from 2023 looking at whole genome level population structuring. Cunner demonstrate very strong population structuring in Atlantic Canada, providing evidence for the last major ingredient needed to test for signatures of adaptation to differing dietary niches. This leads principally to the focus of my PhD surrounding this perfect candidate species. To meaningfully analyze population level adaptation, I needed to address three major hypotheses pertaining to this species using the same set of captured specimens. So first, I needed to ensure that this generalist predator varied in its dietary composition as a function of its local environment that could help promote some regional adaptations. So I investigated dietary composition as a function of region in this species using morphological gut content analysis along alongside DNA metabarcoding. Next, I needed to ensure that these captured specimens still exhibit that same level of population structuring, just to ensure that gene flow was limited and adaptation could be allowed to fully exert its influences. And so whole genome sequencing and population genetic analyses were completed for these same samples. Then, with these two previous hypotheses verified, I'll be able to test for signatures of adaptation within functionally relevant genes pertaining to predatory lifestyles to identify evolutionary processes as they are occurring in this species. So the rest of this presentation will be breaking down each of my thesis chapters pertaining to these three hypotheses, but since the purpose was to analyze the same individuals for every chapter to meaningfully relate the dietary work to the population genetic work to the adaptational work, I need to talk about sample collection first. So samples were collected across much of Atlantic Canada between 2018 and 2022, either by myself or by a previous grad student in our lab. I then collected additional samples from New York in 2024, and this provided a comparative baseline for our northern samples, as this southern region represents an area in which glacial maxima did not reach, thus providing comparison points outside of the identified novel environment. Sample numbers collected for each study in this thesis are presented here, with a total of 125 samples collected that possessed both a GI tract and a fin clip for whole genome sequencing. And these samples sort of overlap with these regions circled here, namely from Nova Scotia as well as from New York for that comparison's sake. We'll begin with my first chapter that looked at the effects of region on dietary composition in this species. So we'll start with some methodologies used for gut content analysis in this species. Specimens were caught and gastrointestinal tracts were removed and preserved on site before transport back to the University of Guelph. Samples underwent traditional morphological analysis involving visual identification of prey taxa. Once this was finished, I homogenized gut contents using liquid nitrogen, then subjected these samples to DNA extraction, PCR amplification, and sequencing using select CO1 barcoding primers. This last part was done largely with the help of Mehrdad Hajibabi and his lab at the Center for Biodiversity Genomics at the University of Guelph. I then subjected these bulk sample sequences to taxonomic assignments using the MetaWorks multi-marker metabarcode pipeline on the compute cluster CEDAR provided by the Digital Research Alliance of Canada. Looking at some results published in a paper in the Journal of Fish Biology in 2025, we have on the left here percent frequency of occurrence of the most commonly identified prey taxa across the study. The top figure here represents our morphological analysis with very broad taxonomic groups, while the bottom represents the metabarcoding results presented at the genus level. We can see across both datasets the strong presence of mussels, small crustaceans, soft-bodied invertebrates like ascidians and bryozoans, which all highlight the generalist benthic predatory nature of this species across its study range. This pie chart on the right represents the results of the morphological analysis, showing the percent weight composition of major dietary items across the study. Again, diet is dominated by these sessile benthic invertebrates like mussels, bryozoans, and ascidians. Moving on to the crux of this chapter, here I begin to visualize differences in dietary composition between our regions. So these figures represent the numbers of exact sequences or total sequence reads of the top 10 most frequently identified genera across the entire study, broken apart by color based on sampling region. We're beginning to see that divergence in composition between certain regions, with certain genera being more commonly associated with samples from specific areas. When I fully investigated differences in composition as a function of region through ordination techniques, I confirmed the existence of differences in composition owing to sample region. These are PCOA plots generated from presence-absence matrices created from our metabarcode detections, species data on the left, genus data on the right. We see clear clustering of samples into three major groups consisting of eastern Newfoundland, southern Newfoundland, and all of Nova Scotia kind of thrown together. Now, this was published before I could actually integrate the results from my New York samples, and so my recent publication that is now in secondary review has integrated these additional results. Shown here are the updated results of this study with New York added in, although it can be hard to distinguish here. Pairwise comparisons of composition between all of the different groups revealed that New York was significantly different from many other comparisons, suggesting that this region also might have differences in dietary composition. When I analyzed only samples for which I have GI tract data and whole genome data, PCOAs revealed the identification of two unique groups, namely Nova Scotia and New York on its own. So we're seeing evidence that the Cunner in this novel northern environment have significantly different diets than their southern counterparts. Next up is the sort of chapter that I only have preliminary data for, but looks at confirming the existence of population structuring within these same Cunner that were captured from my dietary analysis work. For this chapter, I collected fin clips from captured Cunner in the field, subjected them to DNA extractions, and then sent the extracts off for sequencing either to the Center for Applied Genomics at SickKids in 2019 for our Newfoundland and Nova Scotian samples, or to the Advanced Analysis Center at the University of Guelph in 2026 for the New York samples. This led to the corresponding samples and locations on this table on the left. It's from these samples that whole genomes were sequenced and subsequent single nucleotide polymorphism data was analyzed for population structure analysis. Since I just got the raw sequence files back for the New York samples, unfortunately, I can't present any of the results pertaining to that today. What I can show you is the preliminary results for the other 165 samples that I have sequenced. So after some initial filtering of data to ensure only high quality SNPs that were independent of one another were analyzed, I was left with a data set containing roughly 450,000 SNPs for population structure analysis. Following some principal component analysis of these genetic variants and k-means clustering analysis to sort of assign them into groups, I identified 4 unique population clusters that map out onto the respective sampling locations like this. Something of note here is the identification of one large metapopulation across most of Nova Scotia, which matches up nearly perfectly with the identified dietary composition group in my previous chapter. Since I'm working on integrating the New York samples still, this is unfortunately all I have for this work at the moment, but regardless, these results provide evidence to my second hypothesis relating population structure in this very specific sample set. Moving on to my future work that I plan on finishing in the next 2 months, I'll walk through the general methodology that I will employ to test for adaptation in these populations of Cunner. So my previous 2 chapters have now identified A, the presence of unique dietary groups within these samples, highlighting deviation that occurs with sampling region. At the same time, I identified the presence of strong structuring, which nearly mimics the dietary clustering with distinct subpopulations in Newfoundland and a large metapopulation in Nova Scotia. With this in mind, my final chapter of my thesis will look at investigating signatures of adaptation in these populations, specifically in Nova Scotia compared to New York, using the traditional method of SNP outlier analysis. This process usually involves identifying SNPs within regions of the genome that are unusually high in frequencies within certain subpopulations, suggesting adaptive potential of these SNPs or genes. To take this further and fully link all 3 of my chapters together, I plan on using gene ontology searches for identified outlier genes to try to determine a sort of potential functional trait associated with these signatures of adaptation. So looking back at my hypotheses related to my PhD thesis, I've now identified regional differences in dietary composition of this generalist predator within the novel environment of the northwestern Atlantic that may facilitate regional adaptations to prey availability. At the same time, although only a work in progress, I've also identified the presence of significant structuring that may act as a potential barrier to gene flow, further enabling localized adaptation in these populations. And in the coming months, I plan on integrating these results and investigating the potential for adaptive evolution through SNP outlier analysis, fully tying in the results of the previous 2 chapters as a means of investigating adaptive evolution in this species in real time. And this thesis will represent, to my knowledge, the first study integrating whole genome level population genetics with dietary analysis using modern methods like metabarcoding as a means for investigating regional adaptation to predatory niche, providing novel insight into these eco-evolutionary dynamics in a living system. And with that, I would like to thank you all for being here today, as well as to all of the institutions and people that have made this all possible.
Amazing. Thank you so much. So I think we have time for one question. So let's see if we have a question in the Q&A. Okay, we have one. What is the difference in water temperature from New York to Newfoundland?
So it's actually pretty extreme, and they're part of the previous paper that looked at population genetic structuring, kind of related sort of signatures of adaptation in certain populations with sea surface temperature. So it can vary in the summer specifically between 6 to 8 degrees from the southern ranges where we have samples to the farther north in Newfoundland. So it definitely has an impact on adaptation in these species because of that, such a significant decline in temperature.
Thank you so much. And a second one, how deep is the water column?
So they are
Actually, sorry
very
to break up.
they
If we could just take that question to the Q&A.
Exactly.
So
Or
it's
maybe in
time.
the same chat you can answer and
yeah.
think it. So now take it away, Asia.
I was muted, sorry. Thank you guys for coming. My name is Asia Hightower. I am a PhD candidate at Michigan State University in the Joseph's Lab, actually just for one more week. I defend next week, Thursday, so woohoo, go for that. And today I'm going to tell you a little story about some of my final chapter work, looking at how fruit shape relates to reproduction in response to temperature stress in urban weeds. And so if you are at home right now on Zoom and you have any fruits in your kitchen or on your table, you're probably eating fruits from angiosperms. These fruits come from a variety of places across the globe. They include a variety of fruit shapes and sizes and tastes. And one of the cool things about fruit morphology is that it's directly related to differences in plant community composition, evolution, and local adaptation, and also even extinction. And so today we're going to talk a little bit about how fruit shape is related to reproduction, and a little bit about climate change as well. And so the majority of our knowledge from fruit shape comes from crop and model species. And so when we mean fruit shape, we are talking about fruit as like the outline of a fruit, and being able to take a mathematical representation of fruit. This is a really common example in strawberry, where you have these elliptical Fourier descriptors, where you have the outline of the actual strawberry, and it changes across development or depending on the cultivar. And then researchers can work with breeders to pick which cultivar works best and is going to be best for their consumers. So consumers like, a very quintessential strawberry shape. And so this is what we get commonly from crop and model species. However, they're not the only ones that produce fruits. Wild plants produce fruits as well, and urban plants produce fruits as well. And urban plants especially are important because they have unique challenges that we may not necessarily get with our crop species. And these include heat stress due to impervious surfaces and lack of canopy cover. And these plants are really important because they do important ecosystem functions like reducing pollution and reducing flooding and runoff. And so here I'm showing you an example of a heat map in New York, where you see these really strongly hot areas in red, and some of the blue where it's not as warm. And what we know is that for plants in these areas, they have to be able to deal with this heat stress to survive and for their species to persist. And so for studying urban fruit and urban adaptation, Capsulabrus pastoris is a really cool model system. So this is something this is a model system our lab works on really kind of across the lab. And it's a really cool model system because not only is it found in most regions on Earth, everywhere other than Antarctica, it's found in most climate regions, and it has this really charismatic fruit shape. So across the genus, they make this really cool heart-shaped fruit. And they're found in urban areas and disturbed habitats like arable lands. They're cool because you can pick them up on the sides of roads and on the sides of sidewalk cracks and around buildings. So you can kind of find them anywhere. You probably stepped on them outside before. And they have a lot of plasticity in terms of their leaf shape, and also in terms of their fruit shape as well, which I'm going to show you today. They're interesting in that they prefer to overwinter in the cold. So these individuals here in the green, I don't know if you guys can see my mouse, but in the green triangle, you can see where it's between January and about April. This is where they are producing their rosettes. And so they actually prefer between 0 to around 16 to 13 degrees. And then when they're producing flowers, they prefer between 16 to sometimes to 20 degrees, but usually not getting above 25 degrees. So they're cooler temp plants anyway. So they make really great model systems for studying heat stress in these urban environments. Okay. So what we do know about fruit shape for Capsella across the genus actually all comes from studies in one Capsella species, and this is Capsella rubella. So on the left is Arabidopsis salix, and on the left is Capsella rubella fruits. And so you make this quintessential heart shape, like I described before, and we actually call the two parts of the heart valves. So they are mimicking kind of that anthropomorphic human analogy to human shapes. And in the middle, they have this space called a replum, and then you have the style at the top, and then the petiole at the bottom. And you get a difference in shape across flower development where you are growing these kind of more rounded fruits up until they reach this kind of 10-day initiation phase, and then they enter a secondary growth phase, and then you get this final heart-shaped fruit. And so we still don't know for rubella if this is related to fertilization, but we do know that they get these two differences in growth, and it is related to the length of the fruit. And so what we wanted to know was if we grow Capsella pastoris individuals in different temperatures, would we get the same kind of difference in growth and difference in fruit shape? And so today I'm just going to talk about the shape stuff, but if you want to know about any of the growth and the species persistence, I do have a link to our preprint later on. So we grew 5 genotypes, 4 from New York and 1 from Michigan, just to make sure the New York weren't responding that way because they were from New York. In these 3 different temperature conditions in common garden chambers from germination to senescence. We collected a bunch of growth traits throughout their time, and then we dried them in a drying oven for about 2 months. It was really interesting because the fruits at 30 degrees really did not even want to open. The buds didn't want to open. And then they also were green for a really long time. So it took forever to dry. 2 months is a really long time in a drying oven. You don't actually need to dry that long. And then we imaged all of the fruits using a dissecting scope. For our fruits at 16 and 20, they were pretty normal, where they lose their sepals and petals after they are producing seeds. And so you can see that normal valve shape. And then for our fruits at 30 degrees, we actually had to excise them from their sepals and petals. So they stayed closed pretty much the entire time. And then using a leaf shape modified analysis for shape that actually created for a leaf shape for part of my dissertation. We plotted these 2 true landmarks, one at the style and one at the petiole, and then using an automated process, we plotted these equidistant points around the fruits so that we can actually compare across homology, even if we don't actually have all the same biological homology. So we use this to analyze our fruit shapes. And so one of the first questions we asked, here we go, was do we see differences in fruit shape in response to temperature? And we got a very resounding yes, where across this PCA morphal space where you have PC1, PC2, and then projected onto the PC space are these theoretical shapes of fruits based on PC1 and 2. We actually have each point is an individual fruit. And you can see where you get this really strong delineation of fruits that are grown at 16 degrees and then fruits that are grown at 20 degrees, and they have a pretty decent separation with some overlap. And this really different group of fruits grown at 30 degrees. And across PC space, we see that we get this variation in valve width and petiole thickness across PC1, where you go from these pretty wide valves to much more, much smaller, much more succinct valves. And then across PC2, you get this variation in valve depth, where we have kind of our lower end of PC2, these really deep valves with these kind of buried styles. And then as you travel up PC2, you lose that depth and you lose that valve structure to where you're really getting these really rounded valves and you're able to pull out that style in terms of shape. And the second question we wanted to ask was, how is reproduction affected by temperature variation? So we looked at reproduction and fitness across a number of variables, but here I'm just going to talk about the average seed number per fruit. And what we found was that between 16 and 20 degrees, regardless of genotype, there was no difference in seed number. So these are all pretty much the same. But then at 30 degrees, we get this really stark, really kind of like almost startling result where you get no seeds at all. So then it becomes a question of not so much do we get a difference in seed number, but do we have a presence or absence difference in terms of seeds at these in response to temperature variation. And one of the cool things about even being able to look at these fruits through a dissecting scope is that when it's backlit, you can actually see through the fruits themselves because they're really flat. And when they dry out, they're almost see-through. And you can see where at 20 and at 16 degrees, you actually visually can see the seeds. But then at 30 degrees, you can see the lack of seeds. And this looks really like what we saw for those growth phase differences where we're probably getting where if you're not producing seeds, you're not making this this final fruit shape. And so then the last question we wanted to know was what part of shape was actually most responsible for the difference in seed number or the presence or absence of seeds. And so in the red, we have our fruits at 30 degrees. In the blue, we have our fruits at 16 degrees. And then the black, we have our fruits at 20 degrees. And you can see this really strong relationship between length and fruit number, seed number per fruit. And this is also what we saw for Capsella rubella. So it's great to see that we're seeing consistency across the genus. And then when we look at the difference between shape and size, we looked at circularity, which is the ratio of area to perimeter, and then aspect ratio, which is the ratio of width to length. So circularity is how we're looking at shape. And then aspect ratio is how we're looking at size. And you can see these really big difference in these two groups where your fruits that don't produce seeds at 0 are purple, and they're very different than these fruits that produce seeds, which are this orange to pink. So this suggests we're getting this really big difference in developmental programs between the two fruit types, so in response to temperature. So it's really cool to be able to show this kind of direct effect of a change in shape is affecting a change in reproduction. So overall, just to kind of wrap up this little story, we're seeing that fruits in lower temps produce this traditional heart shape. Fruits in lower temps have deeper and wider valves, thinner styles, thinner petioles than the fruits in the higher temps. And that fruit length is strongly associated with the presence or absence of seeds. And then that really the successful fertilization of the seed production is probably necessary to make that heart shape. So that's the next step in some of this work. And with that, I'd like to thank my lab and all of our funding. If you want to contact me, here's my email. And also if you want a link to the preprint, it's here as well. Thank you.
Awesome, Aisha. Thank you so much. And we have 2 minutes for questions. Antoine?
Thank you for your talk. The seeds developing at different temperatures, do they have the same germination rates?
Yeah, so they germinate at about the same rates. They also produce around, so they germinate at the same rates, and they also have the same rate of survival as well. So there is really a difference in the number of individuals in terms of anything in vegetation or for survival, but really it's in the fruit that you start to see differences in the effects of temperature. Hopefully that answered your question.
Is any other question? Or if not, Ryan can share your slides. Yeah.
Thank you so much, Aisha. So if anybody has more questions, you can always put it in the Q&A. And now we're gonna start with our next presentation with Ryan Vu.
There we go. Okay. Well, thanks everybody for attending the talk here. I run the Evolutionary Bioenergetics Lab at Iowa State University, and we study how organisms tolerate stressful environments. We mainly focus on mitochondrial physiology and evolution, and we ask very pointed questions about mitochondrial physiology or evolution, and then we bring in study systems that are best suited to provide answers to those questions, and so we can learn about that. So on the screen here, I've got several of the systems that we work with in my lab, and today I'll focus on a particular question that comes up a lot in our research programs, and that is trying to understand how organisms tolerate bioenergetically challenging environments, specifically related to oxygen availability. So hypoxia, dynamic oxygen availability, things like that. So I'll use springtails here as a case study to investigate their ecophysiology. And the main argument I'll make here is that we have evidence of a horizontal gene transfer event of a mitochondrial targeted gene that's contributing to springtails' bioenergetics and their capacity to occupy below ground habitats. So springtails are an early branching clade of insects, hexapods. They're not insects specifically. Closely related though. They get their namesake from this appendage at the back of their bodies that's called the furcula. Normally it's folded up here underneath their abdomen, and they can fling that furcula open to spring themselves in the air quite acrobatically to escape predation or what have you. The class is separated up into 4 orders, which you can see on the screen here. The scale bar shows that they're quite small and some of them quite adorably cute, like this one here. There's lots of cool biology going on with these springtails besides the spring. One that is interesting is their geographic distribution horizontally. They're found across the globe. But more interesting to me, actually, is their vertical distribution from this above ground to below ground axis. And so the species represented here in these images all represent the above ground species that live in what we call the epidaphic zone. But within most of these orders here, 3 out of the 4, we have species representatives that live across this environmental gradient of above ground to below ground. And this distribution, I think, makes springtails quite a useful model for studying how physiological adaptations might facilitate occupancy of these below ground habitats and the challenges that they bring with them. And there's lots of things that change as we go from this above ground to below ground axis. But as a mitochondrial physiologist, one that I'm interested in is oxygen availability or hypoxia risk, which you can see increases as we go from above to below ground. So this oxygen availability problem is really a mitochondrial problem because mitochondrial processes often rely on oxygen as a terminal electron acceptor for their most famous function of producing large amounts of ATP through oxidative phosphorylation. So I'll go ahead and assume most folks are familiar with this textbook view of the mitochondria and its electron transfer system, where electrons flow through the system and go through what we call the cytochrome pathway through complexes 3 and 4, where the electrons are eventually used to reduce molecular oxygen to water. Okay, so this is how oxygen is used. And this cytochrome pathway is canonical. It's found in all mitochondria. What's less familiar to some folks is this alternative oxidase pathway, or AOX. And here I'll introduce AOX as a potential solution to the dynamic oxygen availability. So it serves a similar function to the cytochrome pathway, where it reduces oxygen to water. It's a terminal oxidase. But the difference is here, when we highlight how hypoxia affects this process. Through nitric oxide regulatory systems, we get a blocking of this complex 4 within the cytochrome pathway. This backs up electron flux through the system and leads to overproduction of reactive oxygen species, or ROS. But in species that have alternative oxidase in addition to the cytochrome pathway, now we have a potential solution to this environmental challenge because AOX is not inhibited by nitric oxide or other commonly found compounds that block the cytochrome pathway. So under this condition, we have a maintenance of electron flux through the mitochondria because of the presence and function of the alternative oxidase enzyme. Now, my lab specializes in this pathway, this AOX pathway. We've recently mapped its distribution across the tree of life from prokaryotes being present in proteobacteria through plants, fungi, and protists. Once we get into animals, it's quite a patchy distribution. But one thing I'll emphasize today is through this work, we discovered potential signatures of horizontal gene transfer of AOX into some animal lineages. So in some rotifer species, in some nematodes, one species of insect, fungus gnat, and then our springtails. Some of these species also have signatures of potential horizontal gene transfer of AOX. Okay, and so the hypothesis I'll be addressing today is that this horizontal gene transfer of AOX into springtails has resulted in this respiratory innovation that can help explain their geographic distribution along that above ground to below ground axis. Now, the first thing we need to address here is the alternative hypothesis to horizontal gene transfer, and that is contamination, right? So I'll do a deep dive into the genomic integration of AOX to robustly show it is consistent with horizontal gene transfer and not contamination. And then we'll go to the functional level and show that it's been physiologically integrated into the springtail repertoire using high-resolution respirometry, and then test for the relevance to the presence of the AOX enzyme to hypoxia tolerance. And then we'll take a macroevolutionary view here and look at the ecological retention of AOX across the phylogeny of springtails using ancestral state reconstruction and phylogenetic comparative methods here for looking at correlations between AOX and hypoxia risk. It's important to recognize and emphasize that not all species of springtails have AOX in their genomes. So a couple of ones that I'll emphasize here in the first part during the experimental section, these two species on the left, the gray and the brown, do have AOX in their genomes, and then the yellow species and the blue lack AOX, okay? So first, squashing the contamination potential here, we're looking at the reference genome of a AOX positive species, and we can see that the AOX gene itself displays typical eukaryotic gene structure. There's 4 exons, the coding sequence is shown in red, and then I've emphasized 4 iron binding sites. Iron is required for the function of this enzyme, and it's strictly conserved motifs at the phylum level or sometimes lower. So here we've got a very short multiple sequence alignment of these 4 iron binding sites. On the bottom here, I've got 2 representative animals, some metazoans, an oyster and a landslip. And then we have our springtail and then a representative Oomycota protist. Okay, so if we look at these iron binding sites, we can see, especially at iron binding site 2, a very clear example where all animal AOX amino acid sequences have this motif at iron binding site 2, mTAL. If we look at the springtail, which is a metazoan, it lacks that metazoan specific motif and instead is more consistent and in fact identical to our Oomycota protist motifs. Okay, so across these iron binding sites and more broadly across the amino acid sequence, we see high levels of similarity between the springtail AOX and the protist AOX, suggesting perhaps this is one of the potential donors of the horizontal gene transfer event. Now, if we look at where AOX resides within the genome, its kind of neighborhood in the genomic context, AOX is in a sea of an otherwise normal springtail genome, where if we blast the genes that are upstream and downstream of AOX on the scaffold, the best hits return other springtail genes and other metazoan genes, right? But only AOX pops up as being more highly similar to a protist than a metazoan. So all of this combined really diminishes the potential for contamination and really supports horizontal gene transfer and full genomic integration following that transfer. But at the genomic level, that's fine. I wanted to know about the physiology here. So I've used high-resolution respirometry of our 4 representative species to test for the functionality of AOX. So here we're looking at the relative amount of oxygen consumption. Remember, mitochondria consume oxygen. And so I won't go into the details here, but our test for AOX arises here in the red box. If we look at the blue and yellow species, which lack AOX in their genome, once we expose the mitochondria from these species to a cytochrome pathway inhibitor, the respiration goes to 0, right? We've blocked the only exit for electrons to consume oxygen, and we've turned off the mitochondria. In the brown and gray species, which have AOX in their genomes, we see a very clear signature of a functional AOX, the persistence of oxygen consumption from their mitochondria in the presence of cytochrome pathway inhibitors. Only when we also add an AOX inhibitor and now block both exits do those species mitochondria fully turn off. We've poked at this in several different ways. One, by pre-treating the mitochondria with one of these inhibitors. So on the left panel of B here, we have pre-treated these species mitochondria with a cytochrome pathway inhibitor, and then given the mitochondria some substrates to try to turn them on. In the blue and yellow species that lack AOX, they never turn on. We've blocked the only exit for their electrons, and their mitochondria are non-functional. The AOX positive species, on the other hand, still respire just fine. And just to show the lack of inhibitor effects, if we pre-treat the mitochondria with an AOX inhibitor, all of the species mitochondria can function just fine. All right, so this really strongly supports a functional AOX that backs up the genomic integration results from earlier. Now, what does this have to do with hypoxia tolerance? Well, I've taken those same species and exposed them to low oxygen concentrations for either 4 hours or 16 hours, let them recover for 24 hours, and then check to see if they're still alive. We can see in our AOX positive species, they recover just fine. However, the AOX negative species really cannot tolerate this short-term or longer-term hypoxia exposure. After 24 hours, they're completely dead or really struggle with the longer-term exposure. And that's consistent with a lack of AOX there. And the cool thing is that these species differ in important and ecologically important ways. Besides just the presence or absence of AOX, they also live across that above ground to below ground axis that tracks with hypoxia exposure risk. So the brown and gray species live either below or in the lower leaf litters or within the soil where we expect high levels of recurrent hypoxia, and that's consistent with the retention or presence of AOX, where the above ground species that have low oxygen, low hypoxia exposure risk lack AOX in their genomes. But these four species, we've seen a correlation here, right, between hypoxia-prone habitat occupancy and the presence of AOX. But it's not just these four species. We see this pattern consistent across the broader phylogeny of springtails. And so this phylogeny is built from Yu et al. Published in 2024. It was built from whole genome sequences of 42 springtail species with some outgroups here. And it's representative of the diversity we see across the four orders. I've mapped on in column 1 here the presence or absence of AOX, absent being gray and black being present. And I've done ancestral state reconstruction to show that at the root of the springtail clade, it's highly consistent with AOX being present at the root of this clade, which is consistent with a single horizontal gene transfer event. So in our present day observations, the species that lack AOX currently lack it because they've lost it along the way of the evolution of this clade. And species that live that have it have retained it since that horizontal gene transfer event. We've also mapped on where these species live in column 2 and their hypoxia exposure risk. And we see through phylogenetic logistic regression that we have a statistical correlation between habitat occupancy of below ground and hypoxia prone habitats with the presence of AOX. So taken together, this all really strongly suggests that we've got a real horizontal gene transfer event of this mitochondrial alternative oxidase that's functional in the springtails and is tracking with this ecological gradient of above ground to below ground habitat occupancy, which is correlated with hypoxia tolerance. So outside of springtails, this broadly represents another example of horizontal gene transfer leading to ecological adaptations through a bioenergetic innovation, which I think is pretty cool. All right, it looks like I'm almost out of time, so I'll go ahead and stop there and take any questions if I have time.
Thank you so much, Ryan. I think we have time for one question. Okay, we have one question. How do you think the HDGT occurred?
Yeah, so in the soil, there's lots of other organisms, including Mycota protists, fungi, bacteria. The ecological opportunities certainly exist within that context, and so it could be just the cohabitation or through eating it or infection. That's a really interesting question that we're not really quite ready to answer, but one that we should look forward to figuring out more in the future. Yeah.
Excellent. Thank you so much. Now we move on into our next speaker, who's Shawn Arreguin.
Hey everybody, you can see my slides okay? Awesome. Thank you guys so much for being here. My name is Shawn. I'm a third year PhD candidate at the University of Illinois, Chicago, and I'll be talking to you guys a little bit about my work. which is trying to understand how urbanization affects reproductive strategies, primarily in this species, Lamium amplexicaule. So why study urbanization? Well, these are really quickly growing environments, both in terms of human population density and in terms of area. And so they are going to be becoming more prevalent. And so I think it's really important that we kind of get to the root of what might be going on, and how that might affect species. In addition, there also are a lot of large-scale environmental changes that come with urbanization, things like pollinator communities, changes to green space, impervious surfaces, habitat connectivity, and also temperature, which I think Asia highlighted really nicely in her presentation. And all these things can kind of culminate into novel selective pressures. And so this is one really interesting to kind of just observe, but also it's a really great opportunity to kind of study these rapid evolutionary changes because these urban areas are coming up at such a quick pace. And this is not a new thing. I'm sure many of you guys have kind of heard about this, but urban evolution has been seen across the tree of life in plants, lizards, birds, just to name a few. And there's a lot of really great examples of this out there. What I'm particularly interested in is how urbanization impacts the reproductive strategies of angiosperms, so flowering plants. So are plants in urban areas selfing more? Are they outcrossing more? Kind of what are those strategies looking like? And so in order to study this, we have two goals. The first is to document phenotypic differences between urban and rural populations in the field, mainly those differences that we can attribute to reproductive strategies. And then two, determine whether these differences, if we find any, are due to plasticity or adaptation. So to do this, we are using Lamium amplexicaule, which is a small weedy mint species. It grows literally all over the place, so it's probably likely that you've seen it, especially if you like gardening. It's a huge garden weed. And it's globally distributed, which is also really great for this work. You know, right now our study is just looking at Chicago, but if we were to ever expand this out of that, it would be pretty easy. And then also within the local landscape, looking at like Chicagoland in northern Illinois, it's well distributed throughout Chicago and then the rural landscape. Like I said, this is also an annual species, and in the greenhouse, it goes from seed to plant death in about 90 days. So it's really great for experimental evolution and documenting evolutionary processes. I just finished up my field season like 2 weeks ago. Pops up in early spring, and it's pretty much dead by now, which is, pretty good. Um, and lastly, this is also a really important aspect for why we chose this species is because of its mating system. It produces two types of flowers, open flowers, also called chasmogamous flowers. I'll probably refer to them as CH flowers. And these flowers are your typical showy flowers that you see on any angiosperm. They can outcross, and in this species, they can also self-reproduce. But this species also produces closed flowers called cleistogamous flowers. These flowers never open up. They pretty much don't get past the bud stage. They're immature buds. And they still set seed, but those seeds are solely self-reproduced seeds. And so using this, we can calculate what's called the CH proportion, which is just the total CH flowers divided by the total flowers. And this is kind of like a proxy for understanding the potential outcrossing rate of, outcrossing rate of this species. And so, for example, if you have a really high CH proportion in one environment, that environment may favor a reproductive strategy that is more outcrossing based. Whereas if you have, very few open flowers, in one environment, then that environment may favor something that is more selfing based. And there are a few things that can affect differences in between urban and rural areas and these reproductive strategies. There are plastic effects such as light, competition, resources, and salt stress. So, for example, individuals in higher light environments tend to produce more open flowers. And we also have selective pressures which are affecting populations over time. So, for example, pollinator communities, if you have a very small non-diverse pollinator community, you may not have selection favoring outcrossing in populations. And so with this, we have two hypotheses for what we might see when we compare urban and rural populations. The first is that with increasing urbanization, CH proportion decreases, so flowers or plants produce less flowers. And in this hypothesis, selfing becomes a more stable reproductive strategy. In hypothesis 2, as urbanization increases, CH proportion increases. So in this case, outcrossing becomes the more stable strategy. So in order to test this, we go out into the field and we find populations of this plant in urban and rural areas. We take a subset of that population and we count the total number of open flowers that those plants produce. And then we harvest those plants at the end of their life and we count the total number of flowers, both open and closed, that they produce. And from here, we can get that CH proportion. So we did this throughout Chicago. We have about 9 sites in Chicagoland. This is for 2025. And then 5 rural sites. I think 2026 was a bit more even. But our preliminary results tell us that as human population density increases, CH proportion decreases. And we used human population density as a proxy for urbanization here. And so we can see that in the scatter plot on the left side. And on the right side, we see a box plot organized by increasing human population density. And so we can see the sites on the left side of the box plot are our rural sites, and they tend to have a higher CH proportion, average CH proportion, but they do have a lot of variation within those sites. Our more urban sites, they tend to have less variation, but also in some cases don't even produce a single open flower. So we're talking about populations that just entirely self-reproduce. So to understand whether these differences we're seeing between urban and rural populations are the result of adaptation or plasticity, we decided to do a very small pilot study in the greenhouse for a little common garden. And so what we did was we took 2 high-flowering mother plants and 4 low-flowering mother plants, and we grew 6 of their self-reproduced offspring from each of those plants in this common garden setting. And so there are 2 things that we might see, one of 2 things we might see for our results. I'll orient ourselves to these graphs. The x-axis is the site where each plant comes from, and the y-axis is the total number of open flowers produced. So each site has 2 dots. So the red dot corresponds to the mother. The blue dot corresponds to the offspring average. And so what we might see in hypothetical scenario 1 is that the mother plant and the offspring produce very similar numbers of open flowers. And this tells us that there is a genetic basis to CH proportion and something heritable going on here. So likely some form of adaptation. In scenario 2, what we are seeing is that offspring across origins, regardless of where they're from, produce similar number of open flowers in this common garden environment. And this indicates to us that environmental variation caused the differences we saw in the field. And so when we actually take a look at our results, we see something that is a little bit more mixed. Now, it does seem that this common garden setting decreases differences in open flower production. So all offspring are producing a somewhat similar number of open flowers, but there still seems to be a lot of family resemblance. So taking this with us, is this adaptation? Is this plasticity? Well, I'd argue it's a little bit of both, that there's a little bit of a genetic basis to CH proportion, but also there's a large amount of environmental variation driving these differences. So some of the things that could be driving this are things like salt levels. So urban areas have a huge salt loads because of our road systems. And that has been shown, salt stress has been shown to decrease open flower production. Also, having large population densities has been shown to increase open flower production. And so that we do see larger populations, at least preliminarily, in rural sites. So from here, what we hope to do is, well, I guess we already finished the field season part 2. I just finished that up. But we did some expanded data collection of the abiotic and biotic factors, such as soil quality and nutrient profiles, and also pollinator communities. And then from here, we're planning to either do a larger common garden experiment or a reciprocal transplant experiment to kind of get to the root of what might be going on, whether it's adaptation or plasticity. I think that's about it. Bunch of people I'd like to acknowledge, including my lab, my committee, the greenhouse staff who have been so awesome, and then all the generous landowners who allowed us to use their land for this work. I think I've got a little bit of time for questions.
Thank you so much. Yes, we do have plenty of time for questions, so please, you can put it in the chat, in the Q&A, or even raise your hands. Okay, we have a question in the chat. Have you considered calculating the FST between the rural and urban flowers?
That is something that we're thinking of doing. We are planning to do some expanded sampling, and then we still have all of our samples from our previous from 2025. So we're hoping to do some sequencing and then calculate some of these population genetic parameters. But yeah, I think that would be really interesting. I
Oh,
think Colette
Colette?
had a question.
Yeah.
I was curious if you have any pollinator observations for any of your populations. I guess that would be another variable that might be contributing to the patterns you see.
Yes, definitely. That could definitely be something. So 2025, I did some initial pollinator surveys, just to kind of like see what's out there. But this year I did expanded surveys, and it doesn't, like, I haven't done any statistics. I really just got this data back. But it doesn't seem like there's a huge difference between urban and rural areas. It seems like hoverflies are the main pollinators. And they seem to be pretty well dispersed between both all of our sites. We do, I think, get a few more bumblebees in rural sites, though. And so that could be something that might be driving it. But yeah, I think that's a really interesting point.
Okay, so if anybody else have more questions, feel free to put it in the Q&A, and we can continue with our next speaker. Thank you, Shawn. Jade. So whenever you're ready, Jade and Solange, remember that this one we're not supposed to be recording.